Hello, everyone. Welcome to this week's Crunch seminar. Thank you so much for your time today. Today, we have two speakers. Our first speaker is Dr. Qing Nie, who is a University of California Presidential Chair and Distinguished Professor of Mathematics and Developmental and Cell Biology at the University of California, Irvine. Dr. Nie is the director of the NSF Simon Center for Multiscale Cell Fate Research, jointly founded by the NSF and the Simons Foundation. In his research, he uses system biology and data-driven methods to study complex biological systems, which focuses on single-cell analysis, multiscale modeling, cellular plasticity, stem cells, embryonic development, and their application to diseases. Dr. Nie has published more than 200 research articles, and in training, he has supervised more than 60 postdocs, fellows, and PhD students, with many of them working in academic institutions. Dr. Nie is a fellow of the American Association of Advancement and Sciences, American Physical Society, Society of Industrial and Applied Mathematics, and American Mathematical Society. And I think that the first talk will be split in two parts. So the second part of the first talk will be given by Dr. Yutong, who's the second-year postdoctoral fellow at Dr. Qing Nie's group. In 2020, she published the first method that infers intermediate cell states and their transition among multiple cell fates based on one snapshot data. This method is based on a non-negative matrix factorization approach in combination of entropy analysis and multiscale modeling. More recently, Dr. Shah led the effort in developing a method, Gong, that reconstructs high-dimensional PDEs of cell fate by embedding and connecting small numbers of temporal snapshots of datasets. Dr. Qing and Dr. Shah, with this short introduction, whenever you're ready, once again, thank you so much for your time today, and you can start.
Okay, so I just I know I found out most of you would be more interested in the math and the methodology behind it. So basically, I will leave more time to Yutong, right? I will give you a quick overview on something you might or might not know much about the modern biology and the new direction in applied math, which is the high-dimensional data and in the application, the context of biology. So I will not go into the depths of biology, and bear with me, right? Since it's reviewed and all the work I talk about has been published. So, and it's easy to, so let me see how I'm gonna move more. Okay, so the single cell genomics, right? It's a technology started about 10 years ago, and it's a revolution technology. And the bottom line is, let's skip the details. It's now you can get in the gene expressions for all the cells and all the genes. Okay. Now you might look at, so you say, what's the big deal? You know, no, it's a very, very big deal. You know, before people can only get in cells for a pre-selected genes and only a small number of genes. Okay. So with that matrix and the effort in the recent years is all about major effort. It's all about the classifications. The people's goal is to nowadays to sequence the 37 trillion cell of human body and classify and to connect this new technology and from the cells to the prime knowledge. So what we are interested is trying to make this a little bit more dynamics, right? So if I given you a snapshot, and many of you know fluid mechanics, right? This is the water wave, and then this drops, and then you can put it in your fluid mechanics equation to make predictions. Here is, if I give you a snapshot of a matrix, what can you do from here, right? So when you're looking at the cell fate decision, usually there's a kind of a gene marker approach in biology. So what I have seen show you here is a very simple example of two gene translations, right? A gene marked as epithelium, a gene marked as mesenchymal, and in order for one gene turns off to the other gene turns on, and then you have 5 choices, roughly speaking, 5 paths. right? Now, of course, this is extremely simple case based on two-dimensional, simplified two-dimensional version, right? Means your life has only 2 genes, okay? Now, if your life has 3 genes, this becomes much more complicated, okay? So the question is, when you get a high dimension, how are you going to looking at those genes, right? How are you going to look at transitions? So the self-weight decision involving 3 major kind of a process. Is the state transition. That's something most of you guys might be familiar with from a mathematics background. Another is social communication, another is spatial organization. So I'm going to quickly touch some of those points and without giving too much of details, right? So one is, how do you infer transitions in high dimensions? Okay, so the idea is you use dynamic system language to connect the more descriptive language like cells, clusters, transitions to a more mathematical language like particles, attractors, right? And the idea is make it more interpretable. Okay, so now how can you do this? So because the data is a static matrix, so one thing we have done is we fit the data in a dynamic system, and basically to fit in the attractor 1, attractor 2, and the transitions, and then we link them, you know, and in terms of a kind of landscape, right? So I will not describe the method in great details. This is all published work. So the bottom line is this. So at the end of the day, you will be able to generate a landscape on the right. Each dot is a cell. Then you basically have a 2-dimensional view of a high-dimensional self-weight transitions. Okay, those are different type of cells. So the mathematically, it just using diffusion map. I'm pretty sure most, you guys knows about these techniques. Okay, so I'm not gonna go into details. And our innovation is about how to fit it. Okay, we fit this data in a multi-scale way. We co-screen. We at the attractor level, we also identify cell location relative to our attractors, and then somehow we fit the probability of transitions. Okay, so if you're interested in terms of the methodology, the algorithm, it's all in the papers. So with that information, you can either look at the transitions. This is all the kind of landscape description. This numbers it's the probability. It's like a 20.9% probability is making transitions from this particular state to that particular state. And then you can doing kind of a multi-scale reductions. It means instead of looking at individual cells, you can look at a group of cells. Okay. And then you can show there's some strong consistency between the group of cells and the individual cells. And then you can looking at the blood and the transitions. So this is the complicated cell lineage for people who knows the biology. Right. All right. So once we published this and people start, I just want to give you one example, how useful is such a method. And this method published in the middle of the COVID years, and then a lab working on human lung cells and used our method to study the cell lineage. It means one cell becomes another cell. This is a human lung cells. Then they found that use our method, they found that there's one cell state which is highly transient. Nobody has observed before. It means could go into many different directions. And then they validated this by experiment, then they published in Nature, right? So recently, they told me they are working on how this transient cell responds to COVID. Apparently, this transient cell is very sensitive to COVID virus, okay? All right, so it means with our method, you can identify the previously uncovered transient information. So mathematically, for some of you might be interested is diffusion map connected with the overdamped Langevin equation, that's well established, but we established kind of a core screen model of the probability of the transition with the more continuum model, which is the transition rate, right? But by connecting to the barrier height. All right, so the data you need for that kind of a method is just one matrix. Basically, we're assuming the system is more ergodic, means one snapshot allow you to observe everything. Okay, now that's not the case if you have a multiple temporal data sets during early development. Means each snapshot potentially have new cell types. Okay, so that's the method Yutong gonna dive into today more in the second half of the seminars. So once you have the network, once you have the landscape, the next question is how the gene are regulated with each other, connected with each other to regulate the transitions. We have a model and to figure out what are the genes that drive the transitions. So basically, it's one state could be a more disease state, and another one could be a more healthy state, and how what are the critical genes can be manipulated to make such transitions. Now, for some of you might know, the genomics world has multiple different type of data, right? So one kind of data is the data I talk about is about gene expressions. It means it's a very kind of in many language is order zero. It's like if you think about the biology, it's a function. And this is like if you do tail expansion, this is the zero order representation of the biology, which is gene expressions. It's the most important one. And the next order one could be, there's many next order, right? Is the chromatin accessibility, which tells you who regulates what, who tells you what are the potential regulation spot. It's a potential. This is a necessary, kind of a necessary condition, not sufficient, right? Those two data types have quite different characteristics. One is more binary, and one is more continuous, right? So to integrate, it's not easy, and we have a way to integrate them. And then once you integrate them, you can do machine learning to build a model to looking at which location, okay, of the chromatin to has a potential regulation, means correlated, or means somewhat of causal, a kind of a weak causal link to the other part of the genes. Then you can establish kind of cis regulations. Okay, so I'm not going to give you the mathematical details, and the method, it's from mathematical perspective, it's relatively standard. Okay, so next one is I quickly touch through transitions and what the gene, how to establish gene connections to quantify the transitions. The next, I'm going to say how cells communicate. At the end of the day, when you're given a piece of tissue, a human tissues, your lung, your liver, the different cells talk to each other. Okay, and then the center cells can talk to the downstream, talk to other cells through diffusive molecules and the receptors. Okay, so can we use, for example, this one single cell data matrix to infer it? Yes, you can, right? Basically, you can use gene expression to estimate the protein level. You can use the protein level to estimate the interactions of two species. You use the interaction of two species to estimate the probability of potential interactions. The ligand receptor interactions could involve in regulators, right? Positive and negative regulators. And of course, is also related to the number of cells who has those, who express those genes, right? So use all of those information, you can compute something called PIJ, which is cell group I talk with cell group J, okay? And then we published this about 3 years ago, and then many people like to use it. It's one of the most highly cited paper in this area, or maybe the whole single cell world. And during the last 3 years, it has more than 2,500 citations. So now so if you're more interested in more recent version, we have a protocol paper, and which is going to be appear very soon. So what you're getting out of this? You're getting out of this is like this. It's just looking at the bottom corner. It's you can have different cell types, right? And then you can looking at the each link. It is a communication of, for this case, it's a particular gene cell communication pathway called VEGF. Okay, this is the old mouse, young mouse only healing. You know, from here you can easily see what you can easily see the old mouse, the aged mouse has more communications actually compared to the young mouse, even though it is known functionally the old mouse only healing is not as good. Okay, so basically this is a very intuitive way to understand the differences between two different pathological conditions or two different biological conditions of the cell-cell communication networks. Okay, all right. So now, you know, the neuron, so I don't know many people here work on neuron. The neuron talks something slightly differently. They have a neuron specific transmitters, and then we develop the neuron chat, which is the analog of the cell chat, but solely focused on neuron specific communications. People really like to use it. We also use developed X founder. Basically, it's a way to say if you only measure, if your data is only come from this piece of tissue, skin, which is epidermal, are you able to infer what the external, then this dermal part here could be considered the external of this epidermal part? Are you able to figure out the receiving signals, okay, from your data, which is limited only on one piece of tissue? That's always the case. No matter what you do, you have to cut one piece of tissue, right? So this is the cell communication is an area which grow very fast. You know, this is a review paper doing about 2 months ago, 2 to 3 months ago, and you can see they did a very nice job, you know, to organize the existing work in a different context. You know, for example, cell chat is one of the major method, and it's a core method, and then a few other our methods also showing up here. Okay, if you're interested in this is very good reviews. So now let's go to the space. I'm going to just quickly touch the space in the next 2 to 3 minutes. I don't want to take away Yutong's time. So our original motivation about 4 years ago is this, right? You have this matrix data, which is static. And then you might also have an imaging data, which has spatial, temporal, even temporal information. For this image, no temporal information. Spatial information, but only 1 or 2 genes, right? So basically you have 20,000 genes, which are human, and then you have 2 genes. The question is, can you connect them? Okay. Now, in principle, if you have enough data from the individual spatial imaging, you can make the requirement, right? Like basically, it is just an optimization problem. And then you add them all those together, basically it's optimal transport. Okay, I don't want to get into the stuff that's all published about 4 years ago now. This method, no, when we did it, we were the very first to link such kind of spatial imaging with the genomics data. Now it becomes a very popular method. This paper is also very highly cited. And then with this, you can connect the spatial information with the non-spatial data. And right, so basically for people who knows the Drosophila, you can see this is early embryo Drosophila, and this is the zebrafish. Even though you have only 47 genes in space, but you have the genomics data, regular single cell data, you can connect them, you can look at the communication flows, you can look at this, right? Nowadays, so now the technology really advances. So no. The technology can say, I can do all the stuff, means I can measure all the genes in space, even though I might not have single cell resolution. And then for that, we developed a method called COMET, and to infer all the spatial communications, in particular with the focus to see how to eliminate some competitive species. Basically, it's optimal transfer map has values, has infinite values at some of locations. Basically, it's to prevent the to prevent the transport, right? So we use like different technologies for people who are familiar with, like very popular technology like VSIMs, SlideSig. So then you can construct cellular communication in space, and very nicely based on predefined, pre-given, based on knowledge database of ligand receptors. Now, if you don't have knowledge-based ligand receptors, you say, I want to look at a more general pattern. You can think about this imaging problem, but it's like in high dimension, right? So if the imaging problem, you can do spatial graph. But the difference between the spatial graph of a typical imaging is, typical images, you have 3 components here. Here is you have 20,000 genes. Okay, so it's kind of a really high dimensional images. And then how you do it? And then one idea we have is we come up with the concept of a pseudo-spatial-temporal map. Okay, and basically this particular map you're looking at, basically the color you're looking at, it can give you a kind of unbiased information about kind of flow in cellular transitions. Okay, so I will not get into the biology, but I just want to give you flavor. So this is a very typical graph neural network approach. And then very recently, you will see the paper soon in the Nature methods. Now, in the very first method I mentioned, it's a multi-scale method, looking at the transitions in the landscape. Now we move that concept in space. And so now you're looking at what you have, this arrow is in space, okay? It's a spatial data. It's no longer UMAP, no longer low dimensional representations, okay? So this gives you a much more comprehensive information about spatial temporal transitions. Okay, so I basically quickly touched, potential kind of so this is called AI. You have AI similar. I feel some of the things we're doing is also AI. It's kind of an AI approach, right? I touched a few methods, most on transitions, and some on cellular communication, and then also looking at the spatial integrations, the spatial information integrations. All right, so with that, I think I'm just going to thank my team, okay? And thank you for your attention. Do you have any questions?
Thank you so much, Dr. Ching. Yeah, do you have any questions before we move to the second part of the first talk?
Hi, I'm Nikhil. I'm PhD student working with Professor Kardena. I have a question. So you mentioned about the multi-omics approach, right, in your slides?
Yes.
How do you fuse the data from different sources you have, right?
Good questions, right? So, okay, that's a data integration, right? There's many approaches, For example, you can do encoding and decoding to the similar low dimensional space with constraint. That's a very popular approach. Okay, so people has more than 10 papers has gone to that directions in the last 2 to 3 years. And but we do differently. We do local aggregations. We use non-negative factorization to learning both two different types together to look at the shared similarities. So there's many different ways of doing that.
I see. All right. Thanks. That was just in general question. Great. Thank you.
Thanks, Nikhil. Do we have any other question? It looks like we do not, so maybe we can start with Yutong.
So can you see my screen?
Yes, we can see your screen now.
Okay, thank you. So I will introduce more about our recent approach called Taiga, how we are going to integrate deep learning and mathematical modeling to generate the interpretable model for the time series data. So at each time point, the cell is in a particular state with some genes turned on and others turned off. And those cells will not, cell states are not going to be fixed. It will continuously change from one state to another one in the 20,000 dimensional gene expression states. And the cell state transitions is actually at the core of biology. For example, it will help us to understand how do we go from one cell to a whole organism, and how the cells will change in disease, and even more, how do the treatment will point the cell back to the healthy cell states. And with the technology of the single cell RNA sequencing, we can take the molecular snapshots of cells in the expression space. Well, during the sequencing, the cell will be destroyed. That means we cannot measure the same cell at different time points. We cannot track the dynamics of gene expression levels within the individual cell over the biological time. So instead, we only have still photos or like unpaired snapshots sampled from the continuous cellular dynamics. And we do not have the direct correspondence between the cells at different time points from the experiments. So then we're thinking, can we come up with an approach to reconstruct the temporal dynamics and to infer the underlying mechanisms from those time series snapshots? And it's quite challenging, like how we are going to describe such continuous dynamics. So here we are going to use a PDE model to describe these complex cellular dynamics, and we will use optimal transport to find out the shortest path for the cells during the cell state transitions. And also the raw data, we know it is around 20,000 genes. It is very high dimensional data. And it is very sparse and include a lot of noise and even batch effects between the different experiments. So here we are going to use deep learning to do data integration and dimensional reduction. And also we will use deep learning as a solver for our data-driven model. So I will start with a brief introduction to the optimal transport and to show you how we are going to use optimal transport to link the different snapshots. So the optimal transport is actually an optimization problem. It helps us to figure out how can we move a bunch of things from one location to another. And in the single cell biology, the cells sequence at each time point is a set of samples drawn from the distribution at that specific time. And by given multiple snapshots of the cells at different time points, the optimal transport will help us to fill in the gaps and infer the most likely path the cell will take across the expression space. So the optimal transport here will allow us to transfer those discrete snapshots into a continuous model to help us to have a better understanding of the continuous cell state transition process. And actually, the optimal transport is widely applicable in various domains beyond the single cell analysis, including the generative modelings and the image processing. For example, the potential flow generator with L2 optimal transport regularity can figure out the transport from the input distribution to the target one with a minimum transport cost. And also, optimal transport can be applied to tasks like finding the mapping between two image distributions, such as the transformation between two cat images. Then how we are going to apply the optimal transport to our time series single cell RNA sequencing data. And the most intuitive or straightforward way is to use the Kantorowicz formulation. So it considers two distributions, the initial one and target one. We are going to find the map function between the two distributions. And also the two distributions are bounded with total mass equals 1. And finding such a map is actually underdeterminate, and it's quite essential for us to choose a map that minimizes the transport cost. So that's where we have the LP Wasserstein distance to measure the cost and identify the optimal mapping between the two distributions. And actually, this formulation has been applied to the time series single cell RNA sequencing data. So it helps to establish the correspondence between the cellular distribution at different time points. So when we are given a cell state at a particular time point, the optimal transport will help us to figure out what are the ancestors and descendants of that particular cell and help us to find the correspondence between the different snapshots. Well, the problem is this formulation only provides a linear interpolation between the two consecutive time points, and the temporal dependencies is missing in this model. And we know that the cellular dynamics is a continuous process, and we are thinking, is there a way to, is there a continuous model to describe such a continuous process? Then we think about the dynamic optimal transport. So it is a reset of the control formulation in a fluid mechanism framework. They used continuity equation to describe the density field and the velocity field. And in order to find the optimal transport, they derived the equivalent form of the L2 Wasserstein distance, which is equivalent to the informal of the energy required to transport the mass across the velocity field. The next question is, can we directly apply the dynamic optimal transport to our time series, the single cell data? Well, it may not be that appropriate. It's because the cellular dynamics is a continuous process with population change. For example, during the embryonic development, the stem cell will divide. And also when the cells reaches fully differentiated cell types, such as our skin cells or blood cells, the cell will have higher potential to die. So in biological systems, the cell proliferation and death will cause the natural variations in the mass over the time. And both the dynamic OT and OT require the strict mass conservation in the model. So this may not be appropriate to describe the biological systems. Then we think about, can we find a way to relax the constraint of the strict mass conservation? So it turns out that we can introduce a source term on the right-hand side of the continuity equation, so that it will help us to better describe the varying mass distributions due to the cell proliferation and death during the biological systems. And this model is underdeterminate, and we use the Wasserstein-Fischer-Rho distance to minimize the energy for the transport cost and to help us to find the shortest path of cells during the cellular dynamics. And here is an overview of our method. It's called Taigang, and we combine the deep learning with mathematical modeling and combine it with unbalanced dynamic optimal transport. So the input of a model will be a time series single cell RNA sequencing data, and we will use self-supervised learning to do the data integration and dimensional reduction. And on the latent space, we will build a model to describe the cellular dynamics and solve it by a deep learning approach. And here are the output of our approach, and I will introduce it more I will introduce those in details later. So let's take a look at our approach step by step. So first, we now understand the cellular dynamics is a very complex process. It involved the gene expression level changes, the cell state changes, as well as the population changes. And here we use a PDE model to describe such complex process. The X here will denote the gene expression state. The V here will denote the cell transition velocity. The rho will denote the cell population density, and that should match the sequence, the snapshots. The G here denotes the growth rate changes. For example, when G is positive, that means the cell will have higher potential to divide. And when the G is negative, that means the cell will have higher potential to die. And we use Wasserstein-Fischer-Rao distance to help us to figure out the shortest path of the cell when they transport those mass during the cellular dynamics. And this, then it becomes an optimization problem. And in order to solve it, we notice that it includes a high dimensional integral. And in the original gene expression space, the dimension is around 20,000. And even we use some supervised learning, self-supervised learning approach to do the dimensional reduction, the latent space is also has high dimensionality, like usually around 10. And it is very computationally inefficient to solve such high dimensional integral. And how we are going to solve this problem? We come up with two-step solutions. First, we are going to derive a dimensionless formulation for our optimization problem. And then we are going to solve it by using a deep learning solver. So when we take a look on our PDE model, we notice that there is a convection term. So it reminds us of using the characteristic lines to redefine the derivative of x to be a function of time t. And by using the characteristic line, we can transfer the original PDE into a system of ODEs. And even more, by checking the trajectories of individual cells, we can further transform the Wasserstein distance into the expectation form. And within this form, we can use Monte Carlo sampling to do the estimation. And there is no need to solve the high dimensional integral anymore. And at the same time, we relax the constraints on the target distributions by adding a regularization for the reconstruction arrow here. And this is our new object function. The next question is how we are going to solve this object function. And if we go back to our original question, we are given a time series data, and we are going to solve the data-driven model from the time series data. And it reminds us of the neural ODEs. The neural ODEs actually use a neural network to learn the derivative of the hidden state and from the time series data. And it's sometimes easier to learn the right-hand side of the equation by learning, instead of learning the solutions directly. And in our dimensionless form, we do have a system of ODEs. Well, the difference is we have a partial information of the form of the right-hand side of ODE. So here we modify the neural ODE a little bit. We use two networks to learn the velocity field and growth rate respectively, and then to use them to describe our density function. And then our optimization problem is solved by the neural ODEs and backward propagation here. And in more details, this is a detailed workflow of our approach. The input will be a time series snapshot, and each snapshot will include a group of cells sampled from the intrinsic distributions. And after we have done the data integration and the dimensional reduction on the latent space, we will first reconstruct those densities from a bunch of samples. And during the training step, we will use two neural networks to learn the velocity field and growth rate. And in the training process, for example, we will sample a group of cells at a one time point and to do the backward and forward integrals to estimate the densities at that sampled time point. And we know that the error will accumulate during the long time integral. So we consider both the short-term and long-term reconstruction errors here in our loss function. And we also add the matrix for our Wasserstein-Fischer-Roll distance here into our loss function. And for the output of our approach, there are two direct output from the networks. One is a velocity field, one is a growth rate. So the velocity field will tell us how the cell state will changes in the gene expression space. And by taking integral of it, we can further infer the trajectories of individual cells, like what are the ancestors and descendants of the particular cell at a given one time point. And because of the neural networks, we can further infer how the genes will contribute to the velocity changes and the growth rate changes, and which we call the gene regulatory network and growth related genes. So basically, it will help us to infer how the cell state happens and what regulate those cell state transitions. Okay, so far we have a reasonable framework. The next question is, how can we test our approach? If we go back to the single cell RNA sequencing data, there is no ground truth how the cell state happens, how the cell state transition happens, and we have no idea what regulate such transitions. So how can we know the inference of our approach is can actually describe the real biological systems? So in order to test our approach, we build a mathematical model to mimic the single cell state transitions. And for example, the model we build here consider 3 cell states and determined by the 3 genes A, B, and C here. And those 3 genes interact in a form of gene regulatory network. It includes like inhibitions and self-activations here. And in this model, it includes 3 cell states. One is the quiescent state. And the quiescent state is quite important in biological systems to maintain the tissue homostatistics. And there is a transitioning state where the cells starting from the high A expression levels will transition to the high B expression levels. And the gene B here will upregulate cell division, which means the cell with high B expression levels will have higher potential to divide. So the transitioning group will include more and more cells. And we use this model as our benchmark, where we do know the ground truth of how the cell state happens and its underlying mechanisms. So we take 5 snapshots of our simulated model and use it as input for our master Taigon. And it turns out that the Taigon successfully capture the trajectories and the regulatory networks. And we also quantify approach when compared to the other existing trajectory inference and gene regulatory network inference approaches. And turns out that our method has better performance compared to the state-of-the-art approaches here. So far, we benchmarked our method using a simulated data. Then let's play with the real data sets. Okay, so here is one example of the time series data. It includes 3 time points. And in this 2-dimensional visualization, you can see that each dot here represents 1 cell. And its color denotes the sequence time points. And you can see the inferred velocity and the trajectories. Well, an interesting part of this data set is it not only measures the gene expression levels of individual cells, it also uses a clonal barcode to track the clonal information of the cells. It means the cells sharing the same clone barcodes are descendants of the same cell at day 0. So this clonal information provides a partial information for the trajectories or the cell state transitions. And we notice that these biological systems, the cell will eventually differentiate into two distinct cell phases. And when we take a look at the day 2 cells and to check the clones, the clone fate probabilities of which is a proportion of their descendants cell communicating to the two different cell phases. And when we look at the day 2 cells clonal fate probability, we notice that even at the day 2, when the cell still sharing a lot of similarities in the gene expression stage, they already have the biased cell fate at such early stage. And our approach is the only one to capture such early fate bias. So, with the actual clonal information, the inferred cell fate predictions from our approach align well with the experimental prior knowledge. Then let's look at like more data set. So here is another data set, which is epithelial to mesenchymal transition. So the cell we are starting from E state to M state, and it is a 5 time point data set. And you can see that after day 3, most of cells already reached the M state. So the inferred velocity here actually match this situation. Well, the majority of cells will have a very small speed. And you can see the velocity inferenced match the temporal information. And we compared another popular approach to infer the velocity field, where they use the ratio of unspliced mRNA here to infer the instant cell state transition velocities. But you can tell that these inferred velocities are quite inconsistent with the biological time. So this is because this approach integrates the time series data into just one snapshot and does not include the temporal information inside this model.
Yutong, can I ask you a question?
Mm-hmm.
Here, when you say like the consistent directions as observed will be like the dots with the circle around it, right? And the predicted is like the field dots. So this, but I cannot see the, for example, in the observed, I don't see any dots that are like empty inside. So that means that your model capture all of them.
I'm sorry, you mean empty dot inside the empty velocity inside the dot?
For example, look here. Look here what I'm writing here.
Okay.
The
Oh, yeah, good question. Yeah.
Yeah.
So yes, there are two different dots. The empty one actually shows the training data, and those dots shows the estimated data. So it's a little bit different.
Yeah, but in this plot we cannot see the training data, right?
Yeah,
We
exactly. I included in the next figure here.
Oh, okay.
Okay, right here. So now
Yeah.
the empty dot show the training data we input into the model, and the dot will show the inferred cell trajectories from the method. So it's just to set the difference between the two cells.
Okay,
Thank you for asking. And before that, we only take the velocity field and trajectory inference on the latent space. And the more exciting part of our approach is we can infer the velocity field and the single cell trajectories back to the original gene expression space. So this is because we use self-supervised models such as autoencoder here. And with the projection back to the original gene expression space, we can provide more data at the unsequenced time points, and to provide the data point at a denser time point. And so far, we only talk about the velocity-related inference. What about the growth rate inference? And for this E to M transition, we notice that the inferred growth is pretty high during the intermediate cell stage. And these align well with the prior knowledge, where the intermediate cell state showing the higher stemness and higher potential to divide. And we compare the other growth rate inference approaches, where they based on some database to figure out the growth-related genes and to compute such growth rate. And you can see that if I use two different databases, it will induce inconsistent growth rates. So again, it shows that our approach provide an unbiased way to infer the growth rate. And further on, we can check the underlying mechanisms, like how the gene-gene interactions will induce such velocity field and how the different genes are going to contribute to the growth rate changes. And turns out that some of the inference align well with the prior knowledge. And further on, we can even check the cell-cell communications at a denser time point. Okay. And then, so finally, we summarized our method and to compare it with other state-of-art trajectory inference methods for the single-cell RNA sequencing data. We can say that currently, only few approaches consider the temporal information into their model. And our approach is the only unbiased way to infer the growth rate. Well, the most exciting part is we combine the deep learning with mathematical modeling so that we provide the interpretability to the model. We can not just know how the cell state transition happens, we can even know what regulates such cell state transitions. And this is the unique advantage by combining the mathematical modeling and deep learning to provide an interpretable model. Okay. And I would like to thank my advisor, Dr. Chinye, and all my colleagues in NIA Lab. Thank you for your attention.
Thank you so much, Yutong. This was a really nice talk. Do we have any questions from the audience? In that case, I have a question. Can you go back to the proposed framework when you had like this latent space and then you were applying your solve?
Yeah. Right here?
Here, yeah, this one works. The first question would be like when you moved the images on the left to your latent space, you said you use self-supervised learning, but when you mean that, you mean like an autoencoder? And if it was an autoencoder, what type of autoencoder did you use?
So here we just use the general autoencoder here.
Okay, and how did you choose the latent dimension?
Yeah, that's a good question actually. So we test using different hyperparameters and to check the reconstruction errors of the different autoencoders with different hyperparameters. And when we say there is and we check how the different hyperparameters well related with the reconstruction errors. If we see that there is no much increase when we increase the number of the hidden layers, then we will choose that one as our hyperparameter, the number of hidden layers. So that's one of the way we choose the initial hyperparameters for the autoencoder. And also we check the output, like the velocity field and the growth rate, back to the original gene expression space, and to check the consistencies of that inference. And that shows that actually, when we go back to the gene expression space, the result shows a higher consistency, regardless of it's not sensitive to the hyperparameter chosen in the autoencoder.
Okay, that's quite good. And also maybe one question here, like, how do you make sure that your PDEs is still holding the latent space?
I'm sorry, how
can you make sure that your PDE still holds or like is still valid in the latent space? Because when you put it from the, let's say, the normal space into the latent space, you mess it up, right?
Yeah. That's I think that's a quite challenging question. So Actually, we do not for this such high dimensional data and to learn the cell state transitions, we do not know the intrinsic dimension of such cell state transitions. We have no idea in advance. So I would say, we use one latent space and to use the PD on that latent space to describe this process, and we have no ground truth of it. So the only way we can test is to check the velocity field and growth rate back to the original gene expression states and to check the consistencies so far. So that's the one of the way to test it, whether we choose the right dimension or right latent space. Another way is we check the output with the literature to check either it match well with the prior knowledge. So we use those two approaches to validate the truth of latent space. Yeah.
Okay.
Yeah, thank you. I think it's a very challenging question.
Okay, that's good. Do you have any other questions from the audience? Okay, it looks like we do not, so let me ask one more question. Why, in the case of your neural network, what you have on the right, your B and your G have the same inputs, right? Like in your neural OD. Why do you need two neural networks? Why didn't you use a single neural network with two outputs?
Yeah. I think yes, we can use one simple neural network. But we use two different neural networks is because we do not want those two to have a lot of influence on each other during the training. And usually we consider like the growth rate can be described by a simpler form of the neural network, while the velocity is maybe it requires more complex neural networks. So in order to based on this kind of intuitive idea, we separate them into two neural networks. And yes, that's the reason.
Oh, okay, I understand. Okay, great. Thank you so much, Yutong and Dr. Ching. This was a really nice talk. Thank you so much for sharing your results with us. You got rid of some really amazing things you did. Yeah.
Thank you.
In that case, I think, do we have any other questions from the audience? It looks like we do not, so maybe we should move to the second speaker. Once again, thank you so much for your time, and it was really great. Dimitris, can you try sharing your screen, please? Yeah.
Can you see it?
Great, that's awesome.
Okay.
Okay, we have as I mentioned before, we had two speakers today. The second speaker is Dr. Dimitrios Patsatzis. I'm sorry if I'm not pronouncing it right.
Okay.
Well, he's a postdoctoral researcher specializing in model reduction and manifold learning within the field of applied mathematics. He obtained his PhD from the National Technical University of Athens in Greece under the supervision of Professor Dimitrios Patsatzis. His doctoral research centered on employing perturbation theory and time-scale analysis to understand and develop reduced order models for complex biological systems characterized by multiple timescales. Currently, Dr. Patsatzis is part of Professor Konstantinos Sieto's research team at the Scuola Superiore Meridionale in Italy. And his current research involves developing physics-informed machine learning methods for numerical analyzing and controlling the macroscopic dynamics in agent-based models, with applications in traffic modeling, crowd dynamics, and system biologies. Dimitrios, with this short introduction, thank you so much for your time today, and you can start whenever you are ready.
Thank you for the introduction and for the invitation on presenting in this great series of seminar. Today I'm gonna present you about how a method that we have developed for discovering slow invariant manifolds that arise frequently in fast, slow dynamical systems using physics-informed neural networks. So let me first provide an introduction of the physical phenomena that we want to describe. So as we know, nature's phenomena are modeled by high-dimensional, complex, and multi-scale dynamical systems. And if we follow bottom-up approach, starting by modeling the individual behavior, like the cars here, or the agents of a market stock, for example, usually we're not able to see the behavior at a collective level. We need exhaustive computations in order to make projections. And of course, it's difficult to track down state transitions and control the system. However, luckily for us, there is an emergent behavior that emerges if you want a collective dynamic or slow dynamics that evolves in low dimensional spaces, as for example here, when you can see that the density of the cars or the change of a stock market as a result of the action of each individual. And we want to actually understand this collective behavior, also build reduced order models to make predictions and accelerate computations. And at the end of the day, perform system level tasks, for example, bifurcation analysis, control design, or others. So given that, we know that every physical phenomenon is multi-scale in nature, but what I want to make here the distinction between the spatial scales and the time scales that exist in every phenomenon that we are studying. By spatial scales, I mean, for example, in this traffic model, when we start at the microscopic level and the atomistic level, let's say you model a car, we go to the road, you model the density, for example, then you go to a larger macroscopic network, let's say. But here in this presentation, I'm going to talk about the timescales that are frequently evolving in such phenomena. So as for example, we have in a classical watch, we have very little gears evolving too fast inside the watch. We cannot see them. And what we actually observe, it's a slow dynamics in the scale of minutes or hours in our watch. So I will follow the next outline. I will first introduce what fast, slow dynamical systems are for a mathematical point of view. And then I will spare a few minutes regarding the geometric singular perturbation theory. That is the theory we study these systems, introduce the slow invariant manifolds and some of their properties, which I will exploit, in order to build a framework that actually identifies these low-dimensional spaces, given a system of an explicit timescale with an explicit timescale splitting, and how we then generalized it in general fast-slow systems without explicit timescale splitting. So the motivation of this work, as I told you at the beginning, the focus of this work is actually in OD systems, which are characterized by multiple timescales operating in similar level of spatial scales. We can find such systems in systems biology and pharmacokinetics, such as the glycolysis path that you see here. In most cases in reactive flows, when we have chemical timescales operating in slow and fast timescales, but also in neurochemistry, in this example here, where you have different agents, different types of neurons, and you have reactions inside, processes inside and outside of the cell operating in different scales. So essentially, we have this kind of problems whenever stiffness is present to our mathematical model. And the physical reasoning for that is because we have many processes, multiple processes operating in their own time scale, together forming a complex system. And by that, I mean that imagine, for example, for the example I gave about the watch, we have a gear turning fast here, a gear turning fast around here, but we only see a behavior, a collective behavior, and that's what we observe. For example, we have processes that evolve to timescales lower than 1 second, and we have others that evolve very slowly. And actually, our objective for this work is to identify the underlying low-dimensional structures that evolve in such kind of systems, which in this case are called slow invariant manifolds, and actually derive machine learning functionals in order to facilitate ROM construction. So, starting with an OD system of this form, where Z is my state vector of dimension n, and F is the vector field F. Now, at the first part of the presentation, I will talk about systems with explicit timescale splitting. So imagine that we have processes involving in multiple timescales, and we're able to characterize them in such a way so that the fast timescale, we can find the fast timescale and the slow timescale, and actually find the ratio, which is below 1 character, and we talk is you can always find this. You can also find this as a perturbation parameter, epsilon. Now, an example of this would be a 2D boundary layer problem. You see the ODEs here. So depending on your initial conditions on the left boundary, you can see a fast evolution here, while then, which also depends on epsilon, the perturbation parameter, while a slow one as time passes. Of course, you may see mixed stuff like this evolution here. You may see a fast oscillation or a slow reduction of the amplitude. But what those systems have in common is that actually they're both fast slow systems. We call them singularly perturbed systems because we can identify, we assume actually that we can identify a parameter epsilon, which is actually expressing the explicit timescale splitting. And of course, as this parameter goes to 0, we can see different qualitative behavior. Now, because of the identification of this epsilon, we are able to decompose the state vector z in two parts, in a fast part and a slow part, x and y, of dimension m and dimension n minus m respectively, and of course define from the vector field to define the fast vector field and the slow vector field. And in this way, we can write the original system in this form, for example, that operates on a fast timescale. This describes the behavior here on the left boundary in this example, or the slow subsystem, which operates in the slow timescale and describes this evolution here. Of course, from the ROM point of view, we're interested in this system because we want to find models, reduced order models for describing the slow evolution at the end of the day. So starting with that, initially what was done before the 80s, asymptotic analysis was used to describe this kind of systems. But later on, geometric singular perturbation theory was established by the seminal work on Fenichel and Tikhonov towards the end of the 70s, which focuses more on the geometric structures that emerge in the phase space and their properties. One of those is the critical manifold of the fast variables. So assuming we have epsilon equal to 0, this is the set of points where the fast vector field goes to 0. And with the assumption of normal hyperbolicity, we can also derive a compact normal hyperbolic submanifold, which is a submanifold of this critical manifold. Essentially, this is a mapping between the slow variables y and the fast variables x. But this is for epsilon going to 0. Now, in the case where the epsilon is small but non-zero, then we have what I initially told you about, the title of my presentation, a slow invariant manifold developing, which is actually a mapping of this parameter epsilon and the slow variables y to the fast variables x. It has a whole bunch of good properties. Its order epsilon close to S0 here. Its flow converges to S0 and has the same stability properties. And the reason why we call it slow invariant manifold is because it's a surface, a low-dimensional surface arising in the phase space, where initially we have a fast evolution of a system, and then after the transit, after the healing, if you may, the trajectory evolves on the manifold slowly. So that's why it's a slow invariant manifold. Of course, this could be attracting on repelling. In the case of attractive, we may have evolution towards a fixed point, towards a limit cycle, or even changing the dimension. If it's repelling, it may for some time evolve on that and then go off. And of course, we're interested in finding these structures because then the evolution on the manifold is expressed by a ROM. So given that we find the mapping of the slow invariant manifold from y and epsilon to x, then essentially we have on our hands the reduced order model, because I remind you that here we have the slow vector field, and if we know the mapping from y and epsilon to x, we can substitute here and get ordinary differential equations for the evolution of the slow variable. So essentially the reduced order model. So the goal, after all, is to compute the slow invariant manifold functional. And I'm going to stress out here the invariance property of the SIM in time, which, for example, imagine that we we search to find this functional. If we differentiate this in time, with epsilon being invariant in time as well, then we end up with this partial differential equation, which is the invariance equation. Here you have the fast vector field, the slow vector field, the functional that you seek to find in the gradient, and inside here as well. And actually, you want to find this functional H to satisfy this invariance equation. So what was initially done was, because of course it's very difficult to solve such an equation analytically, let's say that we write an asymptotic expansion of this, right? And then term by term, zeroth term, first order term, second order term, we try to find these terms. Okay, there is a bunch of methods that they have done that, but those methods all suffer by the same limitation that this expansion is, of course, valid locally for small values of epsilon. And as the number of equation increases, as the number of x increases, the dimension of x increases, these analytic computations or even numerical ones become intractable. To solve some these kind of problems, there are other more sophisticated methods, such as the computational serial perturbation or the ILDM method, which essentially what they're trying to do is, as the trajectory of the system evolves, they define fast and slow subspaces of the tangent space, and then that's how they derive, using the vectors that span those subspaces, they derive the functional of the manifold. But notice here that this functional is implicit in form. It has both x and y inside, and this is an obstacle when we want to derive a reduced order model, because you need to do an extra step. You have to solve for x in order to substitute here in the vector and the slow vector field to get the Rho. So what actually the objective of our analysis, summarized in this slide, is, given a singular perturbed dynamical system of this form, with an explicit timescale splitting characterized by epsilon, we want to compute a sim functional of this form, an explicit sim functional, in a given domain omega for y's and I for epsilon, that is valid for most of the values of epsilon. That it's not how to say, it's not it does not suffer for the magnitude of epsilon that other methods do. If it's in an explicit form, so that we can directly plug it in here and get a reduced order model. And of course, it's easily generalizable, does not suffer essentially from complicated calculations. So the one possible answer to do that is to use a machine learning functional, for example, a neural network, to find this functional, the sim functional. So what actually one could do is to plug in the values of the slow variables of y from 1 to n minus 1 as an input, and epsilon as an input, and match it with an output of the fast variables, the values of the fast variables. This way it's easy to generalize. It's an explicit functional, as I said, and it's not a regular perturbation, a regular asymptotic expansion. These are the parameters of the neural network that you have. And then you just solve a regression problem. You can try to minimize this loss function. But actually, is this a manifold? It would only be a manifold if you were able to collect data for both the fast x and slow variables in order to plug them here, because you need targets to do the regression task. But then you have to make sure that all these data actually lie on the manifold, because then your functional is not a slow invariant manifold. So in order to solve this problem, which in practice you cannot get values for the fast variables because you cannot measure them with the current equipment that we have for experiments, for example, one answer is, one possible thinking was to actually, why don't we make our neural network satisfy a physical law? So that's where the physics-informed neural networks come into play. So what we want to do is actually take a machine learning machinery, neural networks that is trained in order to satisfy a physical law. This will give us better extrapolation properties than regression, and of course will not require label data, the x's that I told you before, the fast variables, to lie on the sim. So essentially what we do is we collect collocation points for the slow variables y and epsilon. We plug them into the neural as inputs to our neural network. We get the output of the neural network, and we want this to be a slow invariant manifold. How will we do it? By actually training it in order to satisfy the invariance equation I told you in the beginning. Right? We want the output of this network to satisfy this equation. So we make this equation a loss function, plug it here as you see. And while the function that we want to estimate h here is the output of our neural network. Okay, so as you can see, we have here the neural network in the output of the neural network in the fast vector field, the slow vector field, and here is the gradient of the our neural network. Of course, we can compute this using symbolic differentiation. These guys we know here from our analytic system. And notice in this framework, before I go into further details about solving it, notice that in comparison to regression, we only require data for the slow variables y and epsilon, and we do not require data to lie on the sim, right? We only need collocation points. We don't want actually to have observations about the fast variables. So we did this with two types of neural networks. We did it with single layer feed-forward neural networks and with random projection neural networks. With the SLFNNs, this is the functional that we used. The parameters we need to find are the output weights and biases and input ones. Imagine that we have here m outputs of the neural network in total. So these are the parameters that we need to find. And the loss function, the optimization problem, now if you consider it, reduces to the minimization of these residuals. Okay, so for the ny and n epsilon collocation points that I have, then I need to minimize these residuals that are m in number, m times ny and epsilon, and find the parameters that minimize these residuals. One could do this with various ways. In our case, we selected the Lamberт-Marquardt algorithm, which is shown here. The most computationally demanding step is this one, the calculation of the Jacobian. And actually, the reason why we selected single layer feed-forward networks is because we could do it with symbolic differentiation and accelerate computations. And for comparison, we also did it with finite differences and automatic differentiation. But even with that, single layer feed-forward neural networks are computationally expensive. And that's why we also introduced random projection neural networks as a low computational cost alternative for SLFNNs. So in this case, we have a similar structure with SLFNS, but now we are randomly fixing the internal weights and biases. Okay, and we only need to calculate the output weights. We set also the output bias as 0, so that we have a lesser number of parameters to find in this case. This kind of neural networks, they have the great potential. They have a universal approximation ability. In practice, there are a lot of methodologies in order to select those guys here parsimoniously to have a better convergence. And at the end of the day, what we care is the less parameters we need to learn. And actually, as I will show you, we do not need to employ the Lamberт-Marquardt algorithm, which is computationally expensive. So recall the first optimization, the physics-informed neural network optimization problem I talked about, the invariance equation. Now the residuals take this form. For every collocation point, we have m residuals to minimize with respect only to the output weights. And now, instead of using Lambert-Marquardt, we can use Newton iteration with SVD in a low-rank Jacobian, actually. And in this step, actually, we're able to employ symbolic differentiation. And because of that, and symbolic differentiation and the use of Newton iterations with SVD, we expect that when we solve with RPNNs, it will be much faster, the training, than that with respect to SLFNNs. So before going to examples, I would like to show you how we collected the data. So given a singular perturbed system in this form that I talked about, we first specify the domain of the slow variables omega and epsilon and I of epsilon we want to compute the slow invariant manifold for. But then, as I told you about, to find the collocation point is tricky, because if, for example, let's say that here we have one fast variable and two slow ones, we cannot just go here and discretize this space and take collocation points, because actually the manifold may not exist for this example here, for this collocation point. So what we did is we selected random initial conditions, generated trajectories in the domain we are interested to, and actually selected equidistant, in this case, time points, and actually collected training and validation sets only for the slow variables, only for y and z in this case, in this domain, for given epsilons that we discretized in space. And then we trained and validated the neural networks with respect to the residual norms that I told you before. Now, in order to assess our pin sim, we did exactly the same thing, but now we waited a little bit for the system to go on the manifold. And then, except from taking only the slow variables and epsilon that we were taking before, we now sampling the ground truth, let's say, numerical solution for the fast variables. And we do that because we want to assess the error of our numerical sim. Okay. So the first example that I'm going to show is the simplest mechanism. It's a mechanism of enzyme action. It's called Michaelis-Menten mechanism. It's the simplest singular perturbed system that you can find in literature. It's a two-dimensional system. It's multi-scale almost everywhere in the parameter space. And actually, we took the most cited singular perturbed form that we have in literature. This is from Siegel and Slemrod. Here, x is the fast variable, y is the slow variable. We have two parameters, k and kappa and sigma. And here is how the slow invariant manifold looks like for different values of epsilon. We have the slow variable here. You can see that the trajectories quickly go to the slow invariant manifold and then evolve on it towards reaching the fixed point, which in this case is 0, 0. As you can see, this is quasi-linear. This is linear in this domain here. So it's pretty easy to find this with analytical methods. And actually, we did that with what classic theory said, geometric singular perturbation theory. We obtained this in a regular asymptotic expansion form. This is the first order, the zeroth order term, the so-called quasi-steady state approximation, first order epsilon term, order epsilon squared term. And then we also did with the more sophisticated methods that I talked before, in particular with CSP. We were lucky in this case. This was an explicit functional. So we collected training data by starting from 10 different, 10 random initial conditions. As I told you, I selected this domain and collected equidistant in time points over here, and for 13 points on epsilon. For SLFNNs, I used 20 neurons in the hidden layer, while for RPNNs, I used 81 because I wanted to have the same number of parameters to find. And these are the results of the training process for all those sims. Now, with this is the SLFNN using automatic differentiation, finite differences, and symbolic differentiation, and these are the results for RPNNs with symbolic differentiation. As you can see, both all the sims converge to very low values of the loss function. But what I want you to concentrate here is that SLFNNs with symbolic differentiation are 3 times faster than with finite differences, which in turn is 2 times faster than automatic differentiation. But the RPNNs are 100 times faster than the faster SLFNN sim that we have. Okay, these results are mean values for 100 runs that we employed in total. So this means that RPNNs are very efficient in training. And let's see now the approximation accuracy of those sims. I collected 500 trajectories in the domain I told you before. And now I get the numerical solution for the fast variable after the trajectory has landed on the sim in order to compute the approximation error. Here is a table summarizing the approximation error over all simulations for all values of epsilon. This is L to L infinity NMSE errors. As you can see here, the pin seams provide very high approximation accuracy, especially the ones with symbolic differentiation, and the SLFNNs and RPNNs are similarly accurate. While if you compare it with the classic approaches, the QSSA, order epsilon squared, the CSP that I told you in the beginning, overall we have a better accuracy than almost all the GSPT approximations, all the regular expansions, and even CSP with one iteration. Now, I want to focus on some visualization here because it provides a meaningful example, a meaningful result here. So here I'm plotting the errors for the values of epsilon and the values of the slow variables y for the least accurate GSPT approximation, the QSSA, and the most accurate GSPT order epsilon squared. And this is for our pin sim for SLFNNs and RPNNs. Now, look at this. As epsilon increases, we expect for these approximations to lose error, to lose accuracy, okay, as epsilon gets high. And this is expected because actually they are regular approximation accuracy. Now, on the other hand, our pin approach has almost the same, is not affected actually by the value of epsilon. And another important feature here is that the first GSPT approximations, when they go close to the fixed point, they are more accurate. While they are away from it, while y increases, they are less accurate. While in our case, again, the pin sim provides similar accuracy all over the domain we are interested to. Okay. We've also done this with other examples, but I will skip it. So this is a three-dimensional example of a limit cycle here. We reach the same examples, the same results in terms of efficiency, but also in terms of accuracy. You see, as epsilon increases, we lose accuracy for the classical approximations, while our sim is not affected by that. I wanted just to concentrate a little bit on this mechanism. It's a three-dimensional mechanism that actually has this feature that the trajectories evolve towards the sim and then they degenerate from it, because actually the fixed point is not on the sim of this the sim. Okay. And this means actually that we're reaching a very nice example, a nice result. Again, the efficiency is similar as in the previous examples, but as you can see here, in terms of accuracy, our pin sims can actually beat well the analytic approximations of GSPT order epsilon and order epsilon squared, and have similar accuracy with what CSP does. But if I visualize, again, the least accurate and the most accurate approximations, we will see something strange, except from epsilon, that when epsilon increases, again, we have similar, our sims that are not affected, while the other sims are affected as expected. So here you see that the trajectories, I'll show you the trajectories come like this, and they go out of the sim like that. Okay, and as you can see, they lose accuracy close to the boundary of the sim when they exit the boundary of the sim. Okay, while in our approach, not only we're not affected by epsilon, but we are not affected so much from the boundary of the sim. It means that we have almost similar accuracy all over the domain, irrespectively if we are close to the boundary or not. And a quick summary for this first part here is that we built a physics-informed neural network sim to approximate slow invariant manifolds of singular perturbed systems by exploiting the invariance equation. Now we actually have the vector fields here, and we do not require data. We do not require data to lie on the sim. That would be the case of regression. And actually, we provide an explicit sim functional that is useful for ROMs. Compared to the GSPT methods that have seen that are reported in the literature so far, we provide similar or higher SIM approximation accuracy, and we are not affected by the values of the perturbation parameter, which is very important because in practice, most of the physical systems, this epsilon is not 10 to the minus 4 or 10 to the minus 3, it's close to 10 to the minus 1 or even bigger. And then we wanted to generalize this framework for systems of the fast slow systems where we don't know explicitly the time scale splitting. So let me get a step back and remind you that we started with this system of ODEs, which characterized by the explicit time scale splitting. This means that with intuition or with some estimation from a local estimation from a Jacobian, we could derive this parameter epsilon, and because of that, we were able to go from this system to this one, where these are the fast variables, the slow variables, and because of that, we are deriving the SIM functional with the way I showed you in the previous part. But now imagine that we don't have this. We don't know anything about the explicit, don't know anything about the time scale splitting. We only know that we actually assume that there is an explicit time scale splitting. We don't know it. So we're able to write our system in this form where we don't know what X is or what Y is. We don't know its dimension, and we don't know, of course, this functionality. Okay. So we want to find an slow invariant manifold that intrinsically exists. And it's a mapping from the slow variables to the fast variables. So again, we assume that there are multiple time scales in the system. We don't know the dimension of the fast and the slow variables. We don't know the transformation from the state variables to these fast and slow variables, and not, of course, not the vector fields. We assume that this exists and we want to find it. So imagine that we start with this kind of system. We want to find the SIM functional, where there is a transformation from the state vector to the fast and slow variables that such that there exists a manifold of this kind. Again, we use physics-informed neural networks, and the benefit of that, to do that, is that we can, with the methodology that I will show you, is that in simultaneous way we can learn the fast and slow variables, how the state vector is decomposed to fast and slow variables, and also provide an explicit functional of that form in order to plug into a reduced order model. And of course, because of the machinery of neural networks, this is a generalizable method. So starting from this domain omega that we have our state vectors, let me define the transformation. The transformation from the fast from this z to the fast and slow variables y, x, and y needs to satisfy this slow invariant manifold. So essentially, we need an explicit functional from the slow variables to the fast ones, where we don't know these transformations and we don't know the functional. These are what we seek to find. Of course, this is invariant in time, as we saw in the previous case. So what we do is differentiate in time and get reach an invariance equation of this form, where here we don't know almost anything. We only know this vector field f of z, which is the one of the original system. So we make the assumption that the transformation from of z to x and y is linear. Why we do that? We do this because also GSPT, which is the method that we want to compare, they do this assumption. So they assume that some of the components of the vector field z, say for example z1 and z3, are purely fast and some of them are purely slow. Okay, so here we make this assumption as well. We assume that there are two matrices that map from the state space z to the fast space and the slow space of y's and x's, and then the invariance equation becomes this one. So in this equation, we don't know the transformations, the linear transformations. Here the matrices C and D. We don't know the slow invariant manifold, this functional H, and of course we don't know the dimension of the manifold. So with the approach I will show you now, we start by we start by collecting collocation points. Of course, the first step is to determine the dimension of this of the manifold. So imagine that we have this kind of trajectory here. We want to collect some samples here on the n-dimensional space and define their intrinsic dimension of the manifold. We could do that with manifold learning techniques. But in this case, we use the so-called CSP criterion. This is a criterion that takes advantage of the eigenvalues and eigenvectors of the linearized system. And for every sample that I select, it can tell me immediately how many are the, which are the dimensions of the manifold. So after we do that, after we select some collocation points and define with this criterion a constant number of dimensions of the slow invariant manifold, we essentially define our slow variable transformation and our fast variable transformation, the matrices C and D I told you before. And now we're able to plug in the neural network to the slow variables. We have the slow variables as inputs, and as output, we have the slow invariant manifold functional that we want to calculate. Okay. Now, in this framework, we first train in an autoencoder, if I may, fashion. So essentially, we say that our neural network will want to be an explicit functional. So the output of that will be the output of this one, and we form this loss function, the first loss function. But also, we want this functional to be invariant in time, actually to be a slow invariant manifold. So this is the invariance equation that I told you before. We give it again, we write it again in a loss function. And we add another one. We add some pinning conditions actually for the transformation, because as I told you, what GSPT assumes is that we have purely fast or purely slow variables. So these are what these pinning conditions do, and essentially we train the pin sim in order to minimize this loss function, this overall loss function. I will show you a little more about this omega in a few if I have time. So we plug essentially an SLFNN. We did it with an SLFNN. So we have to compute these parameters, the parameters of the network, and also the parameters of the two matrices. Here are these parameters. And in similar fashion as we did in the previous with an explicit timescale splitting, we form the residuals. These residuals we're minimizing using the Lambda-Marquardt, and also the use of SLFNN enables symbolic differentiation for the derivatives. So that's how we solve our problem. In order to train the sim, we follow this procedure. That's how this omega comes in play. When we select a number of collocation points from our trajectories, because the manifold does not exist for every value of the slow of Z. Actually, initially we train with omega being 1, which means that we give an equal weight to these three loss functions: the explicit function loss, the invariant equation loss, and the pinning condition. But after a while, after we have learned this transformation, we plug omega being 0, and then we refine our neural network, the parameters of our neural network. So before going into the one example that I have to show you here, I would like to give a brief comparison of our pin sim for the sim approximation and what GSPT approximations do. For example, the QSSA approximation, the partial equilibrium, and the CSP. Now, in our case, as I told you, by construction, we have an explicit functional. These guys here, they are not always explicit, as I told you in the beginning of the presentation. In some of the most sophisticated methods, they are implicit, so we cannot use them for ROM construction. We need to do actually an extra step. Here, the advantage of this methodology is that we have only done an assumption regarding the transformation that we only did because we want to compare with the GSPT approximations. While in these guys here, we need to have knowledge about the fast and slow variables and possibly which reactions are fast and which are slow in order to construct sim approximations that are accurate. Okay. And of course, as I told you, for these guys here, we need an extra step, these Newton iterations to define the manifold. So, and what I'm going to show you next, that we tested our SIB in some examples. I will show you one, and we assessed actually the error of the pin SIM using this error, while for the GSPT approximations, we used the GSPT derived functional. When it was explicit, we also performed the Newton iteration step. So I'm going in this more interesting, to my opinion, mechanism. I will only show this one, which actually I told you before when we were studying it from the explicit timescale splitting point of view, that we have a manifold where the solution goes out of the manifold. Actually, when you look at the original system, you have two manifolds. So initially you have the trajectories going onto one manifold, then going out of this M1, and then after that they approach another manifold and then evolve on this manifold towards the fixed point. Okay, they degenerate from the first, they go to the second, and at the end they go to the fixed point, which is this one. So we tried to, we approximated actually with the pin SIM these two manifolds, but before going to that, I just have a list here from what is known from the GSPT point of view. Here are the approximations. These are the assumptions that you do in order to construct them. And I only want to tell that for this manifold, you need actually, because R, the second variable is fast, you expect only these to be accurate, and this one. And for this manifold, you expect these approximations to be accurate because you build them with the assumption that L in this case is fast. And when you don't require any assumptions, which is the case with the CSP with two iterations, what you actually require is a Newton extra step, because these guys are not explicit but implicit. So here are the training and test sets that I used. And these are the training results for both the first manifold and the second manifold. As you see, they converge pretty high. But what it's important here is the transformation that we learned with the pin functionals. So in the first manifold, as I told you, the fast variable is R, and in the second manifold, the fast variable is L. So what we get here for the transformation is, look, C is 0, 1, 0. So if you have C times Z, you immediately get that the fast variable is the second one, so R. In this case, the fast variable is L, and for the slow variables, you get a linear combination of the slow variables, right? From L and RL in this case, and R and RL in this case. So this means that our pin SIM can actually detect which are the fast and which are the slow variables. And here is the approximation accuracy of the SIM functional. Here I have the pin in both manifolds with the metrics I showed you before, and all the other known approximations. It's too much. I just want to point out that in order to have GSPT accurate approximation, you need to know a priori the assumption regarding the fast variable. For example, here in M1, the fast variable is R. So as you see, the accurate approximations are the ones that are constructed with this assumption, QSSA R, CSP R, and also the second iteration and pair. If you want to drop this assumption, you have to go to these guys here, this one, this 3, the CSP with the second iteration, which actually do not require the assumption on the fast variable, but the trade-off is that essentially these guys are implicit, provide implicit approximations of the manifold. So what you see here is what an extra Newton step required. While in our pin SIM, we get similar or even higher accuracy than the most accurate GSPT approximations, and we have an explicit functional with no assumptions required, right? So this is the beauty of this SIM. And to summarize this whole, the next part, actually we extended the previous framework of pins to compute SIM approximations for a general class of fast-slow systems, which would not have any assumption, any knowledge regarding the explicit timescale splitting. And we showed that this SIM can discover both the fast and slow variables and the SIM functional in one step. We demonstrated this in another two case studies. And actually, it was shown that the pin sim robustly discovers the correct transformations. And in comparison to the GSPT, which is actually what we bring into the game, is that we can get similar or higher approximation accuracy, which is not affected by the boundaries of the sim. We do not require any assumption regarding the fast and the slow variables because we compute them. And we actually provide explicit functionals that are plug and play for ROM construction. Now, in the future, we want, of course, to relax the assumption of the linear transformation, because it's very restricting by introducing a nonlinear mapping, for example, of the high dimensional space to the fast and slow variables. This could require a different criterion to define the dimension of the fast and slow variables, and of course, a lifting procedure from the fast and slow space to the high dimensional space. Another possible direction would be to introduce another physical law in order to mimic what CSP and ILDM do in order to define the fast and slow subspaces of the tangent space. And of course, extend this framework to ROM integration and projection for high dimensional spaces, especially used for PDs. With that, I would like to thank my collaborators, Gianluca Fabiani from SSM, Dr. Lucia Russo from Concilio Nazionale delle Ricerche, and Professor Konstantinos Sietos from University of Naples. You can find the first part of this seminar. You can find it in archive, and it's also accepted in SIAM Journal of Scientific Computing, while the second part is under review for the Journal of Applied Dynamical Systems, also published in a preprint form. And thank you for keeping up with me.
Thank you so much, Dimitri. This was a really nice presentation. You got really nice results. Do we have any questions from the audience?
I had a small question. This is Varun from Brown.
Please, Varun.
I had a question about the random projection that you used. So you said that the first layer you initialize randomly, and then don't train that network, don't train that layer. But did you see like any difference if you initialize it like multiple times in your results?
You're talking about the random projection neural networks, right?
Yeah.
Yes, actually performed 100 runs of for every example I reported, differently randomized, actually. And let me tell, before I answer your question, let me tell something that here actually we randomize this, but we randomize them parsimoniously. So what does this mean? It means that when you have, for example, this training data, you select some of these training data randomly, and you need them to be the centers of the functions that you want to minimize. So in our case, we used sigmoid functions. So we want some of these guys to be the inflection points. That's what we call centers of this function. So essentially, what you do is you randomly choose which these centers would be, and you select the variables b in parameters b, so that they are inflection points. And we did not see in the first example, the Michaelis-Menten example, we did not see any differences between the different runs. What was the suffering case was this one, actually, because in order to converge to some similar error with the SLFNNs, we need much more neurons than what SLFNN did. So actually, this is because of the curvature of the manifold here. If you see, the curvature changes in the change of epsilon. So you cannot just take this one and expect a center and centroid here to project something here. You see?
Yeah.
So you need to sample good in this case. If you have, if you have curvatures, you need to do that.
Okay, yeah. Okay. Thank you. Nice
for the
talk.
question. Thank you.
Great. Do we have any other questions from the audience? It looks like we do not. So thank you so much, Dimitrios, for your time. This was a really nice presentation.
Thank you.
Okay, so with this, we thank both of our speakers today. There was a really nice talk, and thank you everyone for your time today. And until the next week, have a great weekend, all of you. Thank you so much.
Bye.